The life cycle of Cryptosporidium is quite interesting. This the sporozoite of Cryptosporidium contains the actually four sporozoites, and when the transmission, the infection takes place to the takes place via the fecal oral route, or when the food or water is contaminated with the oocyst of Cryptosporidium. So when this sporidium, when the oocyst of Cryptosporidium enters into the intestine, it's a sporozoite. There is existment of the oocyst, and the sporozoites penetrate the microvillus brush border of the intestine, and it is converted there into the trophozoites. That trophozoites is again converted into type 1 cytons. That type 1 cytons is again converted into 4 to 8 merozoites. So there is a change in the stages of the sporozoites when it enters into the intestine, like trophozoites and type 1 cytons. Then type 1 cytons and again converted into 4 to 8 merozoites. The 4 to 8 merozoites has now two choices. It can convert either in type 2 cytons, or again it is converted into type 1 cytons. So the repeated cycle of from merozoites to type 1 cytons takes place, and it enormously increase the number of parasites in the intestine. So this 4 to 8 merozoites is again converted into type 2 cytons. Then type 2 cytons is converted finally into the 4 merozoites, and then this merozoites is converted into undifferentiated gametes, then male gametocytes and female gametocytes. Then male gametes converted into male gametes and female gametes. Then male gametes and female gametes, there is a syngamy, they become zygote. Then there is a sporogony. And those zygote is converted into the oocyst. One thing is very important here, that there are two kinds of oocyst produced into the life cycle of Cryptosporidium. First is 80% of the oocyst produced is actually thick-walled oocyst, and 20% of the oocyst produced is thin-walled oocyst. So both kind of oocyst is produced, and in most of the cases thick-walled oocyst come outside in the environment with the feces of the infected animals, and that 20% thin-walled oocyst is it generally remain within the host, and they take part in the auto infection. So I tell you that auto infection is a very important word in case of Cryptosporidium parvum. When there is an auto infection, the intensity of infection within the host, if it keeps on increasing, it means there is no need to pick up infection continuously from the external environment. Once you infected, the parasites keeps on increasing. The intensity of the parasite, the number of the parasite in the host of the in the body of the host keeps on increasing, and the gravity of the infection within the host is increasing as time passes. So when you overall talk about the life cycle of Cryptosporidium, the two things are very important. There is extensive recycling of merozoites from the type 1 cytons, or the auto infection within the host is responsible for the massive infection. So when they read the life cycle, you must remember that there is a continuous recycling of merozoites within the host, and the auto infection leads to the massive infection in the host. The host which comes out from the which comes out from the infected animal is already a sporulated one, so that is the immediately infective to the host. There is no need of there's no need of to be a sporulation, no need of a sporulation, because the oocyst is already uh, fully sporulated. When you talk about the pathogenesis and symptoms related to Cryptosporidium, it depends upon the intake of the oocyst, type of the species of the Cryptosporidium, immune status of the host, and I have already told you that it mainly affect the microvillus brush border of the intestine. So there is a villus atrophy, hyperplasia of the villus, shortening of microvilli, sloughing of the cells. So these are the pathogenesis which takes place in the host. In the case of Cryptosporidium, mainly the young animals, mainly less than 1 month of age, or maximum 1 to 3 months of animals are seems to be mostly affected. The adult animals are also infected, but in their in the adult animals, the infection is basically self-limiting, and after getting well, these adult animals may as a source of infection for the calves. So most of the infections are seen in the calves which are 1 to 3 months of old, and when it comes to the symptom, there is a profuse cholera like diarrhea, abdominal pain, anorexia, result dehydration, malnutrition, malabsorption, and the due to the severe diarrhea there might be dehydration. The animal keeps on losing weight, and if the animal is concomitantly infected with other infections such as E. coli or rotavirus infection, this might then really create a big problem. When it comes to diagnosis of the Cryptosporidium oocyst, there is one important question is asked, that what is the staining method of the oocyst of Cryptosporidium. So acid fast staining is required to see the Cryptosporidium. Actually, the oocyst of Cryptosporidium is one of the smallest oocyst among coccidia, and they are difficult to see in the normal preparation of fishes, because they are very small, they are colorless, they are transparent, non refractile actually. So it is very difficult to see the oocyst under normal microscope actually. If you have the phase contrast microscope, then we can see the oocyst directly. But in most of the cases, if you want to see the oocyst, the we have to stain that we have to stain the oocyst with uh, with several staining techniques like modified Jaden staining, oramin phenol, carbolfuchsin. So in which there are several staining technique which I have already told you, in which the oocysts are generally stained red and counter stain, and there is counter background is counter stained with bluish color. But modified acid fast staining and quinone stain is regarded as the gold standard staining technique for the Cryptosporidium. So I repeat that modified acid fast staining technique and quinone stain is still regarded as the gold standard staining technique. One thing is more here, that if the animal is a carrier, and shading oocyst in a very less amount, in that case it is also difficult to oocyst in a direct fecal smear examination. In that case, we have to concentrate the oocyst. There is one solution which is known as Sheether's sugar solution. I repeat, that one solution that is known as Seether's sugar solution, which is used to concentrate the oocyst of Cryptosporidium, and this is a even we can use the zinc sulfate saturated solution of zinc sulfate to concentrate the oocyst. Both these solutions are used in the flotation technique. Saturated salt solution or saturated sugar solution might deform the oocyst of coccidiosis, so they are not used. The best solution for the concentration is Seether's sugar solution, and this question is generally asked in the examination. When it There are several diagnostic techniques like ELISA, direct fluorescent antibody test, latex agglutination test are available. Even the molecular test, PCR based diagnosis is also available for the diagnosis. We can use primers based on 18S ribosomal RNA, which can not only differentiate the species. When you see the oocyst under the microscope, all oocysts are morphologically same. You cannot differentiate the species on the basis of morphometry of the oocyst. But when it comes to molecular techniques, we can differentiate the Cryptosporidium up to the species level with the use of from different primers. And as far as the prevention and treatment is concerned, I have already told you that oocysts are quite robust one. It can tolerate the extremes of temperature and cold. It can survive up to the 0 degree centigrade and up to the 65 degrees centigrade. A very wide range. 10% formalin is required, or 5% ammonium solution is required to kill the parasites, to kill the oocyst actually. When the treatment is concerned, in case of human, a drug called nitajoxanide is already approved by FDA USA to use in humans. When it comes to animal, there is tylosin, erythromycin can be used, but there is no specific drug or vaccine for the treatment of Cryptosporidium. For the prevention, hygiene is utmost required. Hygiene is required. We have to stop the contamination of our food, and water. So this is all about the Cryptosporidium. If you have any query related to this, you can post me on the Google Classroom. uh, Several questions generally asked. One question which is very pet is which kind of staining is used. The oocyst is various. Which of the following coccidian parasite has very smallest oocyst? So I think that is all about the Cryptosporidium. Thank you.